Hi, my name's Toby Phesse. I'm here, my lab's here in the Cardiff University. Thanks first to AMMF and particularly Helen for inviting me to talk today and tell you about the work that we've been doing investigating how Wnt signaling regulates metastasis in cholangiocarcinoma. So Wnt signaling is a highly conserved pathway. Regulates many cellular functions, including proliferation, differentiation, migration, and cell death. And these are very important features in embryonic development, and indeed Wnt signaling regulates and is critical for embryonic development and indeed homeostasis in several tissues. I've spent about the last 15 to 20 years trying to understand how Wnt signaling regulates many of these functions, and these are just a few pictures out of various papers. And not only does Wnt signaling regulate these cellular function in normal tissue, but it is deregulated in many cancers, and all these different cellular functions, if they're not correctly regulated, can lead to transformation or tumor progression. And so Wnt signaling is of great interest in this regard as a potential target for cancer therapy. So this is what the canonical Wnt signaling pathway looks like. This is a very basic look at it. It's always getting added to almost every day. A new component gets added. It's very tightly regulated. There's also a non-canonical pathway, but we'll just look at the canonical pathway today, not to say that the non-canonical pathway isn't important. Anyway, so in the canonical pathway, the main component here is this co-transcription factor beta-catenin, and its cytoplasmic concentration is being continually regulated by this degradation complex, and they phosphorylate beta-catenin and target it for degradation. Upon the on the receptor on the membrane level here, the Wnt ligands themselves are being regulated by SFRPs, and the Wnt receptors, these frizzles that want to bind to their co-receptors LRP, are being continually taken off the cell surface by this E3 ubiquitin ligase, ZNRF43 ZNRF3 RNF43. So that's the situation when the Wnt pathway is off. When it's on, this E3 ubiquitin ligase is now targeted for degradation itself, because RSPO, which is the ligand for LGR5, binds and then sequesters RNF43 away from frizzled, and it can just get on with its job then of transmitting the signal. The Wnt binds to it, it inhibits this complex, tethers it up onto the membrane, get a buildup of beta-catenin into the nucleus. It can then go into the nucleus and activate its transcriptional targets. So Wnt signaling is deregulated in many cancers, as I mentioned, but it's also deregulated in cholangiocarcinoma. However, if you look at the frequency of mutations, actually incredibly low compared to other cancers like APC, AXIN, beta-catenin, RNF43, very low. I mean, with APC, the levels of mutations in a disease like colon cancer are around 90%. However, if you look at some of the factors that are regulating the pathway at the level of the receptor, and the ligands, you find that these are methylated and therefore silenced in cholangiocarcinoma. And this is where I really got interested in cholangiocarcinoma. This is from a paper that Luke Bolter did in JCI. And this figure here, these are all these Wnt target genes up in human cholangiocarcinoma. And there's one of these in particular, frizzled 7, that I was working on at the time. And indeed, Luke, in his paper, he used these Wnt inhibitors to target the pathway, and he could prevent the growth of the primary cholangiocarcinomas, suggesting then that Wnt signaling is important in regulating this disease. So Wnt signaling also regulates lots of features of metastasis. So as I've just shown you, Wnt signaling can regulate the primary tumor, as Luke just showed in cholangio, but it's also in lots of other cancer types. But it can also regulate EMTs. This is where the tumor cells need to change from a mesothelial to a mesenchymal type, so they can migrate away from the primary and invade either the blood vessels or sometimes, as in cholangiocarcinoma, into the peritoneum. Wnt signaling can promote this. It can also promote survival and control cell death, as they need to survive when they circulate the body. They need to then go back to an epithelial cell type, and we and others have shown that Wnt signaling regulates this MET transition. And then they need to escape dormancy. Often they just lie dormant, but to actually form a metastasis, they need to activate their cancer stem cell properties and form this metastatic tumor. And so Wnt signaling has been shown in other cancers to regulate lots of these features. Cholangiocarcinoma is a highly metastatic disease, so patients often present with advanced disease, and if they do, then the 5-year survival is only 2%. Terrible. Resection is the only curative treatment option, but even in resected patients, recurrence occurs in about 65%. So that just shows you how metastatic this disease is. And secondary tumors mainly develop in the peritoneal cavity, the liver, lung, and bone. And so as Wnt signaling regulates many of these features, we investigated if it was required for metastasis in cholangiocarcinoma. And so the approach we took was to actually inhibit the secretion of Wnt ligands. And this can be done with this drug called LG974, which I'll sometimes refer to as Wnt974. And so the way that Wnts are secreted requires this acetyl transferase called porcupine. And unless it is acetylated, it doesn't go through this process of being secreted. And that's what LG974 does. It inhibits this cell transferase and prevents the secretion of Wnt ligands. So the first thing we did is we used this drug on some cholangiocarcinoma cells, just to show that here we could inhibit Wnt signaling. This is top flash, which was a Wnt reporter assay, and also some endogenous genes in the Wnt pathway. So MYC, cyclin D1, ROC1, which is actually a non-canonical Wnt target gene, and also these two genes, CD19, CD44. These were of interest because they were markers of cancer stem cells. So this suggested then perhaps Wnt signaling might be regulating this process in cholangiocarcinoma cells. And so we performed some colony forming assays. This is where you plate out a very low density of cancer cells and ask them to perform, to grow colonies. Some of them can grow colonies, but only a very small percentage have this property. And if you ask them to do it when they can't secrete Wnt ligands, you can see you can almost completely block their ability to have this cancer stem cell activity. So showing that the cells themselves, this is a cell intrinsic, there's no other cells in there, cell intrinsic mechanism. And to better try and mimic where the cells would be when they actually do this, so obviously we did that on plastic, we did it in matrigel, so they have a bit more anchorage independence, but we also did it in the extracellular matrix from the lung and the liver. And you can see here in these bottom two, you get lots of colony forming in these two extracellular matrix scenarios, mimicking the niche, metastatic niche. And again, you can significantly inhibit their ability to form these colonies and their cancer stem cell ability if you block Wnt secretion. So a feature of metastasis is also the abilities of cells to move. And we picked 3 different cell lines here, 2 from intracellular cholangiocarcinoma, 1 from extracellular cholangiocarcinoma. And in all 3 scenarios, if you inhibit their ability to secrete Wnts, you can block significantly inhibit their ability to migrate as well. And also, as I mentioned, EMT. So the cells need to go from epithelial to a mesenchymal cell type. And you can see we've got this way of doing this in vitro with something called stem ex vivo. And you can see we can transfer these cells from an epithelial to a nice mesenchymal type. And we can also monitor this molecularly with these markers ZEB1, slug, and snail. So these all go up when you have this EMT transition. And also Wnt signals go up. So frizzled, cyclin D1, MYC, TCF, they all go up. And now if you ask the cells to try and undergo this epithelial to mesenchymal transition, without Wnt signal, without Wnt ligands, you can completely block the ability of this transition. And we did that in 2 different cell lines. So as LG-974 blocks the secretion of all Wnts, we next wanted to ask which one of the Wnt receptors was important for this. So Luke's paper in JCI identified that Frizzled-7 was one of the Wnt receptors that was upregulated in cholangiocarcinoma, but there's 10 frizzled receptors. I've spent the last 10 years looking at this particular one, Frizzled-7, and we found that it's really is a key Wnt receptor regulating stem cell function and regeneration, tumor growth in the intestine and the stomach in vivo. So we were interested to see if Frizzled-7 was transmitting Wnt signaling to promote these metastatic functions in cholangiocarcinoma. So this is just some work we previously did in the stomach. So this is a stomach pinned out now, and in this model, you can see that there's these tumors. And if we use this drug, which blocks 5 of the frizzleds, including Frizzled-7, you can significantly inhibit the number and size of these tumors. And similarly, if we now do a genetic deletion of just Frizzled-7 on its own, we can phenocopy this, and we can almost completely block the development of these gastric tumors. And we published this a couple of years ago. So we inhibited just Frizzled-7 with this shRNA, and we did our top flash assay again. So this is just looking at canonical Wnt signaling, and it can block Wnt signaling, not quite as much as the inhibition of with LG-974, which blocks the Wnt secretion. However, when we looked at the endogenous gene expression, we've got just as much knockdown of those genes as we did with LG-974 blocking all secretion, suggesting that Frizzled-7 is again a key Wnt receptor transmitting these Wnt signals in cholangiocarcinoma. And to confirm this, we could overexpress Frizzled-7, so we get good overexpression of Frizzled-7 here, and that's associated with an increase in the expression of Wnt target genes. So what does this do functionally? So we could see that it inhibits the migration of these 2 CC cell lines with the Frizzled-7 shRNA. And conversely, if we overexpress Frizzled-7, we can increase the migration of cholangiocarcinoma cells, and also their ability to form colonies. So their cancer stem cell function is also significantly decreased if you inhibit just one of these 10 receptors, Frizzled-7, suggesting that Frizzled-7 is an important receptor, as we'd shown in the GI tract. So what have we got so far is we've got the Wnt ligand signaling through Frizzled-7 to regulate several of these functions: EMT, migration, and cancer stem cell function, all features of metastasis. So in Luke's paper, he'd identified actually the source of where these Wnt signals were coming from. It's from the macrophages. So we were next interested to see what happens when a cholangiocarcinoma cell actually arrives at its metastatic niche, in this case in the peritoneum. And so to do this, so the peritoneum is actually lined by this mesothelial layer. So we took cells from the mesothelium and grew them in vitro, and then removed the media and replaced it with serum-free media, and then allowed the cells to essentially secrete whatever they were secreting into the media. And then we asked the cells then to migrate in this mesothelial conditioned media, and found that actually they had a significantly higher propensity to migrate towards this conditioned media from the mesothelial metastatic niche. So the next question was, okay, what is this signal that the mesothelial cells are secreting? We've shown that Wnt signaling was important in a cell intrinsic fashion. Is the metastatic niche also secreting Wnt signaling? So this time we actually treated the mesothelial cells themselves with LG-974 to block all the secretion of Wnt ligands. So they're secreting everything else out into their media except Wnt ligands. And then we repeated the experiment, and this time you can see that if you now try and ask these cells to migrate in this conditioned media, which is exactly the same apart from it hasn't got it's Wnt free, and you can completely block their ability to, well, rescue their ability to migrate towards this metastatic niche. So now we have a slight improvement of this now. So we have Wnt ligands being transmitted via Frizzled-7 to promote EMT and migration towards the peritoneal metastatic niche, which is actually secreting the Wnt ligands also. And we have cancer stem cell function. So these then suggest that targeting Wnt signaling using this drug, LG974, is an attractive therapeutic strategy for treating metastatic cholangiocarcinoma. So we next tested this now in vivo, and here we inject the cancer cells into the peritoneum directly, and if you wait 3 or 4 or 5 weeks, you can see we've circled here these metastatic deposits, and we can weigh those, and in this mouse here on the right, which we've treated with LG974, we can almost completely block, and in some cases, so each one of these is a mouse, in some cases completely block the metastatic deposits that can develop in this model. We also noticed that the liver was bigger in this model, and we didn't observe that in the LGK974 treated cohorts. And we could weigh the liver, and you could see that this was indeed a phenotype. And then when we looked into the liver, we noticed there were some deposits. So we did some immunohistochemistry to confirm that these were indeed human cells. This is a human mitochondrial marker. So you can see this nice little sort of dog leg shape here. And this was closely associated with the cells that were proliferating in the liver, probably driving this hepatomegaly. This is Chi67, which marks proliferating cells, and you can see that this is just done on a serial section, but you can see they're obviously the same cells. So that just leaves me to summarize and thank those involved and the people that funded it. So essentially, Wnt signaling regulates key functions, key features, of metastasis, cell intrinsically via Frizzled-7, but Wnt signaling also from the peritoneal metastatic niche, promote migration towards it in a Wnt-dependent manner, and also targeting Wnt signaling with LGK974 can inhibit metastasis to the peritoneum and the liver in vivo. So thanks first to Luke for giving me the cells and kicking this project off. So my other collaborators, this is my lab. The people who did this work were Sarah and Val. The people who were involved with the frizzled work is this is when I had my lab in Australia with Professor Vinken and Dusty Flanagan, who's now just trying to set up his own group. And thank you very much to AMMF for their funding of this project. Without that funding, we couldn't have done any of this work. So thank you very much.